All right, tribe, we did it. Lecturette 7. We are at the end of lecture 9. We are going to be talking about long-term potentiation and depression. This is one of my favorite lectures, and it really sets the stage to teach you what you need to do to succeed in any type of learning because of the mechanisms that I'm about to discuss. Now, I want to kick off this lecturette by doing a comparison. So we just learned about short-term signaling, EPSP, IPSP, and how the summation of these weak signals can have the cell or the neuron then integrate at the axon hillock to decide, hey, do I send this signal and keep propagating it down my axon, or do I do nothing, right? Now, I want to stress that these EPSP and IPSP signals are short-term. They last for milliseconds, okay? This is not long-lasting signaling. And the reason being, if you folks remember, is that the ions are traveling very quickly down their concentration gradients, and then they eventually get sucked out of the cell, right? Because of all the ATP pumps that are trying to maintain the resting potential and gradients of certain ions. So this is short-lived. Now, there is long-term signaling, right? And so long-term signaling is long-term potentiation, which we're going to call LTP, and long-term depression, which is LDT. Now, long-term potentiation is very strong signaling that can last weeks, months, and even your entire lifetime. Is because what you're doing with long-term potentiation is you're actually changing the structures in your head, so much so that you can take a slice of your brain before and after initiating long-term potentiation, and you can actually see more tracks in somebody who has initiated long-term potentiation. Okay, so long-term potentiation is much like going to the gym and lifting heavy weights and causing your muscle morphology to change. You can change your brain morphology. Now, long-term depression, and this is the last I'm going to speak of it. I don't want to talk about it too much because we don't have a lot of time. But long-term depression will undo all of the long-term potentiation. So for example, in long-term potentiation, I just told you that you're going to change the structure of your brain, meaning that you're going to actually make more tracks, right? So let's say that you do all of this hard training of your brain, like you folks are doing now, right? And you build all these tracks, right? Now, guess what? You take a break and you don't keep training. What's going to happen to your brain muscles? They're going to start to go away, right? So you go backwards. That's what long-term depression does. It undoes all the long-term potentiation structural changes in your brain. This is why if you don't use it, you lose it, right? Okay, let's continue on. All right, let's talk about long-term potentiation. So I want to introduce a new word to you, something called neuroplasticity. So in biology, when you see the word plasticity, it doesn't mean plastic. It means the ability to change, right? So our bodies are always changing, right? You want more muscle? Lift heavier weights. You want less muscle? Don't lift heavy weights, right? You can manipulate the way your body looks based on how you train. And your body can respond to different types of environmental pressures because it has what we call plasticity. Now, if we say neuroplasticity, this is the ability for the brain to change, which is a very, very amazing thing. We are born with a number of neurons. Can we grow more neurons? Very little. Okay. Before it used to be thought that we couldn't, the neurons we had was the neurons. That was it. That's all we could have. But the brain can, and it has a limited amount of ability to generate more neurons, but not too much. Okay. This is why when people sustain like a heavy brain injury or even a stroke, if you have a minor stroke, maybe you could build over years of training your brain, you can generate a little bit more neurons, but usually strokes are permanent is because you kill those neurons and you can't generate enough neurons in order for you to regain that mental abilities. However, what the brain can grow in great amounts is more dendrites and branches of axons. And if you think about this anatomically, right? The dendrites and the axons, this is where signaling can occur. And you can grow these. You can grow them and have more and more and more to allow you to have more signaling. Okay? Let's put more arrow. Now, that's neuroplasticity. Now, you can have change in the brain if you are persistent, okay, in strengthening your synapse based on activity. That's why I tell you folks, you need to train for this class in a certain way. You can't just read, right? You can't just read for this class is because the signal's not strong enough. And you can't take a break from this class. I like to say that physiology is a jealous lover. It wants all of your time. Okay? You can't just take a break. You can't take a 5-day break. You're not strengthening the synapses that way. Okay? There's no pattern of signaling. Your body, when you study on a daily basis, it creates signaling to where it shows the brain there's a need. There's a need to make more dendrites and more branches of axons, make more neurotransmitter. And when you give that pressure to the brain, then it starts to respond, much like muscles at the gym. That's why I keep telling my students, why are you doing a powerhouse study on the weekend? You're not changing the morphology of your brain. It's like if I want to go get buff and I go to the gym, I'm like, oh my God, I'm going to just exercise Saturday and Sunday, boom, 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 and come Monday, I'm going to be ripped. Does that happen? No, it takes months of training, weeks of training in order for you to change what your body looks like. Same thing with the brain. Now, creates a long-lasting increase in signal transmission between two neurons, right? If you want to have long-term memory, you need to have communication between neurons, right? This is the recall. Recall a beautiful memory that happened to you folks when you were a child. You can remember that is because the neurons that are storing that information are all still connected up. And when you recall it, those neurons will light up. Okay? Recall something that was very negative from when you were a child. You remember that because both positive situations and negative situations give such strong signaling in the brain that they get ingrained in your hippocampus in long-term memory, and you can recall them because those synapses between those neurons are still intact. Now, there's special ways that you can strengthen a synapse which can allow you to have a larger voltage change. And I'm going to show you folks this voltage change. I'm going to draw it out for you folks. But the larger the voltage change you have, the more strengthening of a synapse you will have. And the more strengthening of a synapse you have, the more dendrites and branches of axons can you grow. Okay? Now, when we do long-term potentiation, in order for me to make more dendrites, make more branches of axons, make more neurotransmitters, I also have to make more proteins because those neurotransmitters have to bind somewhere, right? So when you get LTP signaling, you are synthesizing a lot of new proteins, okay? And this is why it takes weeks, months to grow your brain. And I'm saying grow in quotation marks. You can't see me, but I'm making air quote marks, okay? Now, the key neurotransmitter for long-term potentiation is glutamate. Good old glutamate. Did you folks memorize the neurotransmitters yet? Okay, it's okay. Here, we're going to start together, right? Glutamate. We're going to call each other team glutamate, right? Glutamate is key. if you want to make long-term significant changes in your memory, in how you view the world, you need glutamate as a neurotransmitter. You have to turn on glutamate. And I'm going to talk about how you do that. Okay. And glutamate is key. Now, if you want to remember something, recall on an exam and know it, deep learning and know it, you need to initiate glutamate signaling. Okay. Now, this is why people with drug addiction, it's really hard for them to get off drugs, is because the type of signaling that occurs when you do drugs that feels good, you release so much glutamate that you now make connections between some behavior, right? So let me just show you folks. If this is my neuron, this is neuron 1, and this is talking to neuron 2, and then this is to my arm, and this is the pattern of my arm, that all the skeletal that allows me to lift cigarette to mouth. What happens when I keep lifting the cigarette to my mouth? I keep lighting up this pathway, okay? And guess what? The cigarette makes me feel good. So I get more and more glutamate and dopamine and serotonin, but let's just talk about glutamate. Okay, sorry, I'm getting excited. Sherry, stay on topic. Okay, here we go. The more I smoke a cigarette, the more I strengthen the synapses, right? And I keep using them. And the more it feels good, I start to make more connections to the point where the connections and the proteins and all of these tracks become so much that you could see this if I slice your brain open. If I crack your skull open, I start slicing the area of your brain that's responsible for lifting arm, put cigarette in mouth, I can see these tracks. I can see how much they've grown. This is why addiction is so hard to break. Is because not only do you lose the good feeling, you now have to undo, via long-term depression, undo and erase all of these new proteins that you have made. Okay? This type of brain morphology, this is where I want you folks to go with your studying, where you're creating these tracks so much so that you don't even have to think. You just know. You know the material. You know it inside and out, because these neurons are lighting up so well that the recall just happens. There's no pause. There's no blanking out. That doesn't happen. It's because we build new connections. Okay? This is why I build the class this way. I test you folks on the homeworks with time, right? Because I'm trying to stimulate glutamate production for you all. We do application problems so you can use the material. It's because I'm trying to recruit glutamate signaling for you folks. All this training and how I do everything in this class is for your benefit, is to make sure that glutamate is secreted. Not all of this short-term signaling, right? When you folks read and just go through the slides, that's all EPSP, IPSP. You don't hold on to that very well. If you do, it's all short-term memory. And guess what? Short-term memory, it goes away. You need to get your short-term memory into short-term memory and then shuttle it to long-term memory. And that only happens when you have glutamate signaling that is very strong. Okay, let's get into the mechanism. All right, people, let's do this. This is the mechanism for long-term potentiation. All right, now, what's a neurotransmitter? Glutamate. People, we're going to be called Team Glutamate. I'm going to say Team Glutamate. And that's going to, I don't know, hopefully get your glutamate going. All right, so here we go. Glutamate. Now, glutamate can bind to 2 receptors. So I'm just going to draw them. And the first receptor is AMPA. And the second receptor is NMDA. Okay? So what AMPA is a sodium gated channel, right? So what can happen is glutamate comes because this is an excitatory neurotransmitter. It will bind to both of its receptors, but with AMPA, when it binds, it's going to cause sodium to come into the cell. Okay? And then you already know what happens when you have sodium come into the cell, right? Depolarization. And if you depolarize, and if you depolarize enough, then you can now propagate the signal down the axon. Propagate signal means move the signal forward. Keep it going, okay? Now, here's the thing. If you can secrete even more glutamate, then something special happens. And this is why. Is because look at this NMDA receptor. The NMDA receptor only works in the following two conditions. The first condition is glutamate has to bind. That's binds, not bids. Put an N in there. Thank you. Number two, okay? You have to have strong enough depolarization inside of the cell. Okay, so the NMDA receptor is very special. It needs two things to kick it off, right? I have to have glutamate bind, and only if depolarization is strong enough. And this is all dependent, right? When we say if depolarization is strong enough, this is all dependent on how much or the concentration of glutamate that's released, okay? So the more glutamate I have, the more it binds to AMPA, the more sodium is going to come in, the more depolarization I can get. So I have to have enough depolarization. And if I do, and if I reach threshold, and I'm just going to change my color because there's a lot of different processes that are going on. I'm going to change my color to red. Then and only then can I have calcium entry. Okay? From the NMDA receptor. Oh, calcium. Okay, so calcium is special. So let's talk about what calcium does. And sorry, I'm starting to stutter. It's because I'm getting excited, so I have to do my Lamaze breathing. Okay, let's do this. Okay, so here. You folks already know about calcium, right? Calcium does a whole bunch of stuff. And one of the things that calcium can do is activate some second messengers. Which ones? We're just going to say second messengers for right now, okay? And in activating second messengers, I can now do the following. I can now do this. Oh my God, this is so magical. Okay, here we go. I can now go back and phosphorylate. Remember my notation, P circle with the tail is because I stuck a phosphate group, okay? If I phosphorylate the AMPA receptor, yes, I can keep AMPA open. I can keep AMPA open even when glutamate is gone or not binding. Okay? So think about what I just said. I activate second messengers. I can now come back up here, and I don't want to draw the phosphorylation tail, okay? But I can come back here and phosphorylate AMPA here. And even though glutamate's not binding it, I have now kept it open from the inside of the cell. And if I keep it open, If I keep AMPA open, guess what's going to happen? I'm going to have more sodium come in, more sodium come in, more depolarization, which is going to lead to more calcium entry. Okay? Now, when I activate second messengers, you folks already know that activating second messengers can allow me now to activate gene transcription. Why do I transcribe genes? To make more proteins. What proteins do we need? Oh, let me tell you. I can make more receptors. And the more receptors I make, the more AMPA I make, the more NMDA receptors I make this neuron more sensitive to glutamate, meaning that I can have even stronger glutamate signaling. Here's another thing I can do. I can make more dendrites. And how do I do that? Is via increasing protein synthesis of the cytoskeleton. If I make more cytoskeleton and I make it, I can now stick that cytoskeleton, right? So if this is my neuron, if I start growing a cytoskeleton this way, what's going to happen is my cell, let me show you before and after, my cell is going to start to grow another dendrite because that cytoskeleton is in there pushing my plasma membrane out. So if I want to make more branches of this neuron so I can have more connections between neurons, I make more proteins. I transcribe more cytoskeleton, I can make more dendrites, and guess what I can put on these dendrites? I can put more receptors all over here. I've now increased more surface areas for receptors to reside. Oh my God. Here's the last thing calcium entry is going to do, okay? And this is just ridiculous. Now, ridiculously good. Now, calcium entry. You folks know that calcium allows us to have exocytosis. Yes? Oh, man. I'm smiling. Okay. I can have exocytosis of a paracrine molecule. You folks remember paracrine? Paracrine? Paracrine is a molecule that's going to be released from one cell and bind to the next cell or bind to the next tissue. Okay? I can stimulate a paracrine that will bind. So this binds to the presynaptic cell and causes it to secrete more glutamate. Shut the front door. Can you believe that? That is amazing right there. That is amazing. And this is what we're trying to do for you all in this class by training and studying in a specific way. Is because reading and just watching are very passive. You can't get enough glutamate signaling to facilitate changes in the brain. Okay? So how you train is very, very important for your success in education. I want to end this lecture by showing you folks this article. Okay? And please don't memorize all of this false creb. I just want you folks, just pay attention to what I'm saying. Right? Now, what you folks are seeing on this left side of the picture is I have a neuron with a certain amount of dendrites. And you have to remember is that dendrites and the soma of the neuron is where we receive neurotransmitter signaling, right? It's because there's receptors here. So what you folks are seeing here is a blow up of the dendrite, and you can see little receptors. And look, this is the presynaptic neuron with its axon terminal, right? So synapse and a synapse. That's what's going on here when we have all these connections from neuron to neuron. Now, when you're exposed to drugs, what happens? Look at the picture. What is happening here? This is the strong glutamate signaling. Because what did I just grow? I just started to grow a whole bunch of dendrites. And look at the receptor number. You see how it increased? Yes? This is what happens to the brain with drug abuse. And this is why it is so hard, especially right now. We are in well, there's a lot going on in our world right now, okay? But let me not go there. But we're in an opioid epidemic. There are a lot of people dying from opioids is because they are so hungry for the drug, is because every time they take that drug, they start to stimulate more dendrites and more receptors to the point where if they don't have that opioid, their neurons are screaming for the lack of that ligand binding to this receptor and undergoing major withdrawal. Right? And this is why it's so hard to quit opioids, is because quitting is not enough. It takes time for you to degrade and undergo long-term depression, to degrade all of these dendrites that you've now built up over time, and to degrade all of these receptors that you've built up over time. Okay? All right. I'm going to close there because this was a short lecture, but wow, I just looked at the time and that is enough. Okay, much love to you all. Keep studying. Team Glutamate.